Let's look at some definitions which are associated with. So we have genome sequencing, we have the transcriptome sequencing, which is the mRNA content, Why RNA? How does it differ from DNA? Which is the kind of the mRNA content, which is which is the entire genome, uh, you know, after transcription, right? So you have mRNA. What are the consequences of looking at the transcriptome as opposed to the genome? So you get a notion of alternative gene splicing. You learn about post-transcriptional modifications. Gene fusion, you learn about mutations, SNPs, where something doesn't translate. There's some differences, and change in gene expression from tissue to tissue over time, which is epigenetics, right? Based on kind of influences from the kind of environmental influences. So let's look at some of the definitions associated with some of the words here, in case they are not very clear. So let's look for some of these definitions. So that nature, subject, alternate splicing. Some of the definitions. Again, it's a very useful thing is when you are looking for, you in a bioinformatics, when you come across something very specifically biological, very specific, something that's obvious to an experimental person, if you say, well, what is this? You can go to some of these definitions. So alternative splicing is a process that enables a messenger RNA, an mRNA, to direct synthesis of different protein variants, isoforms, that may have different cellular functions or properties. It occurs by rearranging the pattern of intron and exon that are joined by splicing to alter the mRNA coding system. So it really comes from the same DNA, but there's all these cellular processes that occur, and as these cellular processes occur, that same DNA gives rise to different protein products, and each of these protein products is then used in different function, and it all comes from the same gene. Okay, let's look at another definition. So what is post-transcriptional modification? Post-translational modifications are modifications that occur in proteins catalyzed by enzymes after it's translational. So when the mRNA translates into the protein, where the protein manufacture occurs, there are modifications that occur in the protein. So these modifications refer to the addition of a functional group covalently to a protein, for example, a phosphate group, which is called phosphorylation, netylation, but also refers to proteolytic processing and folding processes necessary for a protein to mature functionally. Okay, so some of these modifications occur and they have direct bearing on the function of the protein. So post-translational modifications. For those of you who will take the class taught by Dr. Masood, which is advanced mass spectrometry related work, you will really see that after you send a protein through a mass spectrometer and you get all these peaks for the fragments, you'll see that some of the peaks are slightly shifted because they are associated with post-translational modifications. What are the definitions? Gene fusion. So what is, and this comes from the National Cancer Institute at NIH, a gene made by joining two different genes. Fusion genes may occur naturally in the body by transfer of DNA between chromosomes. For example, the BCR-ABL gene found in some types of leukemia is a fusion gene. Fusion genes can also be made in the lab by combining genes or parts of genes from the same or different organisms, and you connect them by a linker, something in DNA or protein, which is flexible enough to link the two genes. Okay, mutations and SNPs we've already looked at, and let's just look at a quick definition of epigenetics. So, what is epigenetics? Epigenetics is a study of a biological mechanism that will switch genes on and off. What does that mean? So we understand cells, DNA, sequences, etc. So certain circumstances in life can cause genes to be silenced or overexpressed. In other words, they can be turned off, becoming dormant, or turned on. Epigenetics is everywhere. So again, it's the environmental influences which modify gene function. So for example, what you eat, where you live, who you interact with, when you sleep, how you exercise, even aging can cause chemical modifications around the genes, which will turn the genes on and off. So, so in certain disease such as cancer or Alzheimer's, various genes will be switched in the opposite state away from the normal healthy state. Epigenetics makes us unique. So let's see, even though we are all human, why do some of us have blonde hair or darker skin? Why do some of us hate the taste of mushrooms or eggplants? Why are some of us more sociable than others? The different combinations of genes that are turned on or off is what makes each of us unique. Furthermore, there have been indications that some epigenetic changes can be inherited. Okay, so a correlation between an environmental influence and turning the gene off or on and somehow modifying its functioning or removing its functioning or enhancing its functioning, that kind of a response is epigenetic. Okay, one more, epigenetics is reversible. With 20,000 plus genes, what will be the result of the different combinations of gene being turned on or off? The possible arrangements, obviously, are enormous. But if you could map every single cause and effect of the different combinations, and if we could reverse the gene state to keep the good while eliminating the bad, we could theoretically cure cancer, slow aging, stop obesity, and so much more. And so this is a resource which is be really useful because it is dedicated purely to epigenetics. Little more on RNA-seq. It is used to compare gene expression between conditions such as drug treatment versus non-treated and find out which genes are up or down regulated in each condition, because you're really looking at the proteins, right, at the mRNA. RNA sequencing or RNA-seq will allow you to account for all the transcripts in the cell for each condition. Differently expressed genes can be identified by using tools that count the sequencing reads per gene and compare them between samples. A little more information. So there's these DE-seq and EDGE-R packages from Bioconductor can be used to assess differential expressions. Now, I think Dr. Caleb Phillips from the Department of Biology teaches a course in metagenomics, and Dr. John Dawson from the Nutritional Sciences teaches a course in biostatistics, and both of them in their courses teach you R. And Bioconductor is kind of a module in R, which makes running some of these very specific software related to biostatistics, related to bioinformatics, a lot more easier. Okay, so again, the RNA-seq de novo assembly. Remember, de novo means that you have the reads, you have to put them together in some semblance of a proper order, but you do not have a reference sequence. Okay, so this is a little bit of the tuxedo suite which I had talked about. Bow tie for RNA-seq short read alignment, top hat for aligning reads to a reference genome to discover splice sites. Remember, alternate splicing. Cufflinks assembles the transcripts and compares, merge them with others. And then there is cummerbund. So all these are associated with tuxedo. Is an R-based statistical package, can be used to generate expression comparison charts for visual analysis and etc. So at Johns Hopkins University, you'll be able to access the software. I think all of it is free. Now let's just kind of end this by talking a little bit about de novo assembly. So there's an absence, as I said, the de novo assembly is because of the absence of a reference genome or transcriptome. The reads cannot be aligned against the reference. Now, before I talk about the de novo assembly, I talked about the biostatistics by John Dawson and the metagenomics course by Caleb Phillips. I also want to talk about kind of an advanced genomics course that is taught by Professor Rao Kotapalli at our center. And I think this is a superb course. If you want to join that course, you have to get in really early. Places are limited. But I think that's a great course because it allows you, Dr. Kotapalli and his colleagues will teach you not only the experimental basis for this, but you'll also be using the software and actually running through some of these that you'll also have an experimental view of actually using Illumina for next generation sequencing experiments, but you'll also get an opportunity to work on the software side of things. So do recommend these courses. And the last slide I will have is I will give you information about the 3 courses that you might want to that you might want to, one or more of these courses you might want. And again, like this is so getting into these courses will give you a really complete view of kind of, being at the cutting edge of bioinformatics and biostatistics at this point in time. So going back to de novo assembly, because of the assembly of a reference genome or transcriptome, reads cannot be aligned against a reference. So, well, if you don't know what you're aligning against, how do you know that your assembly is good? And here is an example of a paper which will tell you a little more about some of this. De novo assembly, what every biologist should know, and you should be able to access this through our library. Okay, so this is a very important paper. Just, even if you're not going to do anything with it, just read it, get a good sense of what is there, because I have a feeling, even from a job perspective, if you were really looking at this practically, even from a job perspective, I think this is a great resource, a great paper to begin to understand what's going on. It will also help you to inform your decision as to what other courses you would like to take, which would significantly help you. So from this particular paper, it's one kind of a methodology of how to assemble de novo sequence. Now remember, you fragmented your DNA, whether you've used microbeads, whether you've used the channels for the Illumina, and you've gotten all your reads. These are your reads. Well, you still have to find overlap between the reads, like we talked about earlier, except in this case, you don't have a reference genome. So for example, you have this long read and this long read, and there is this significant overlap in yellow. So you realize that this is really one part of the sequence. And this is the kind of assembly that takes place. And the other thing that you can also do is but remember, there's assembly. So there might be, if you're trying to run a software to go and find this, it might say, well, I found this big area, but then it might say I also found this little area. So the question is, how do you differentiate that what is your primary goal, right? And so remember, you're assembling these reads into context, right? Contiguous sequences. And we've looked at when we looked at the definition of an N50. So let's say, for example, you have various reads, and you see the overlap, and then there's another overlap, and so you look at all these overlaps, and you're able to put these overlapping sequences together. And the software called Velvet is a software which actually uses this. And what we are looking at here is what is called as Tebron's graph. Okay, so a little bit of iffiness with the pronunciation of Dutch names, but I think the correct pronunciation is Tebron's graphs. And so let's look at these graphs, right? So let's say you have, and we'll give you so for example, this is a PowerPoint that you can really look at. It's online, and it'll give you a better idea of this. But let's say, for example, and again, in order to make a big problem small, just so we get a kind of a physical picture of what to do, let us look at these reads. Let's assume these are reads, okay? And these are reads, and you want to see how they coincide with each other. So if you see these reads, you'll see there's an AAG part, and then you realize that there's an AAG part, and then there's an AGA part. And then you look at the AGA part, and you see that there is a GAC part, okay, a GAC part, which comes from here, okay, and then you say, oh, the AG and the AG are the same, right? It could be AG, this AG is the same as this AG or this AG. Then you have a GA, which you might say, ooh, that is coincidental with this GA or this GA. And you see, what you're seeing in this Tebron's graph is there's all kinds of these options, right? So you go this direction, but now this GA can come from the GAC, but it can also come from the CGA. Okay, so that feeds into there. But then you have a CG, which can come from the CCG. Okay, a CCG, and then you say a CCGA, and then you say there is a CC here. So I need to find something where there's something which has a CC which might overlap with it, right? And then I have a CC that comes from here, TCC. Okay, then I say, well, there's a TC here, so can I find something with a TC here? and then I find a CTC here. And this is how you begin to, break these apart, try to put them together, try to see that, well, there's a GA which comes from here, there's a GA which comes from here, there's a GA which comes from here. Okay, and then that's when this is how you put this together. And this is essentially an example, a kind of a simple example of a difference graph. Okay, and so now once you begin to put some of these things together, you can say, oh, look, AAGA, that's great. GACAT, which is great, GACT, because the AC and the AC are the same. And then you see there's a CCGA and the CTT, CTCC, and those can be then put together. Okay, and so this is how you begin to put these together, and this methodology is called the difference graph. And now you can put it up, and then, for example, you can now combine the CCGA with the CTCCGA, see that, and then GACT and CTT. And this is basically an example of difference graph. Now, what is Galaxy? Galaxy is a portal. Okay, I think it's a free access. And what the Galaxy project does is it's a portal that allows you all kinds of very interesting resources that you can use, and you can actually get onto the Galaxy portal, Galaxy resource through the portal, and you can actually run your software there. You can upload data, you can kind of drag and drop and create these assembly lines of various software that you can use, and you can run the software that way. So this is a very good resource if you are interested. And that's about as much as I wanted to say. Again, remember that what we discussed is a state of the art. It's kind of revolutionary. It allows us to really process information at the whole genome. We are very lucky to have a kind of an expert in next generation sequencing in our center. So these are the courses that you might be interested in. Dr. Rao Kotapalli from our center, who teaches a gene expression analysis. That's BTech 5312. Again, you need to get into this class, you need to get in early. Dr. Caleb Phillips teaches metagenomics. Now, this is listed as an undergraduate class, but I know for a fact that 2 of my students last year have taken it. Very important course. You'll get introduced to R programming. And Dr. John Dawson teaches NS 5342, which is biostatistics and nutrition, and you get a chance to learn R. Very important courses which are kind of slightly downstream from what is more. This is more of an applications methodology course, okay? But these courses delve deep, and especially Dr. Kotapalli's course and Dr. Phillips's course will really go into the genomic aspects of bioinformatics, which I think is going to be very, very useful for you.